Thank you. Thanks
Thanks.
again.
Thanks, Leah. Yeah, no worries. Okay. Sorry, everybody. I didn't get a chance to practice this, but we'll just wing it. So nice to be back speaking to the Tick Academy. The last time I presented to the Tick Academy, I presented some of my work on how fire and fire management impacts ticks. And I'll be sort of what I think of presenting as the flip side of that coin, which is how invasive plants and invasive plant species alter tick-borne disease dynamics. So this is a major area of focus of research in my lab, and I'll be presenting some of my own work on this as well as work of colleagues. This has been a very active area of study just for about 15 years now. And so you're looking at an image of some non-native invasive plant species. These are barberry shrubs in the background here, and you'll see them again in just a little bit. So I like to start off by presenting distributions of tick species and their associated diseases in the US, because while certain ticks and certain pathogens receive a lot of attention, there's actually a lot more going on. So I'm showing in this map here distribution of tick-borne disease cases in humans reported to the Centers for Disease Control and Prevention in 2015. And you can see a couple things that emerge from this. One is that the eastern half of the US has a very high prevalence of tick-borne disease in humans. But even within the eastern US, there are different clusters of different tick-borne diseases. So Lyme disease receives the most attention, and it's certainly the most prevalent tick-borne disease in North America. But we have other diseases transmitted by various tick species that can cause serious illness in humans, including Rocky Mountain spotted fever, anaplasmosis, ehrlichiosis, babesiosis, tularemia. These are all bacterial and protozoal diseases. We're also discovering pretty rapidly that there are some viruses transmitted by ticks in the US as well. They seem to be less common, but they can be quite serious. We have a number of different vector tick species. The black-legged tick, which is the primary vector of Lyme disease in the eastern US, gets a lot of attention. A tick that I'll actually talk about quite a bit today, the Lone Star tick, is thought of as an important emerging vector of several diseases. The American dog tick is also widespread. We're seeing expanding distribution of the Gulf Coast tick in the southeastern US. And then, not to neglect the western states where there are also tick-borne diseases, another species of Ixodes, the western black-legged tick, is the main vector of Lyme disease in the western US, and the Rocky Mountain wood tick is an important vector of Rocky Mountain spotted fever. So we have a number of different tick species, a number of different pathogens associated with them, and the distributions of many of these ticks and their associated pathogens are changing. And so we're seeing increasing geographic distribution of a number of these diseases expanding into new areas. Overlapping with this is that we're also experiencing widespread invasions by non-native plant species. Many of you likely have encountered these yourselves during your travels in nature or in more developed areas. Just a couple examples on the left there, stiltgrass in the upper image, bush honeysuckle in the lower image. These non-native species that are successful invaders likely have some traits in common that allow them to be able to invade new places, new ecosystems, and displace native species. They often, when they're highly successful, form what we sometimes refer to as monocultures, like these images on the left, where they basically replace all the native vegetation in the understory. And invasive shrubs like honeysuckle actually are killing forests in the sense that they're preventing recruitment of new trees. So young trees are not able to grow up through this canopy of bush honeysuckle. And so there are places in the US where forests are actually going to die because of these invasions. Again, not only is the eastern US very ticky, it's also got a really high density of invasive plants. And so looking at forest inventories performed by the USDA, there are places in the eastern US where even a majority of the understory is composed of by non-native plant species. So we have plant invasions occurring concurrently with ticks spreading to new areas. And, I'm maybe getting just a bit ahead of myself here, but I'm going to show you today a number of examples of how invasive plants seem to be impacting ticks and their associated pathogens. And one of the big questions I'm interested in is, are these plant invasions also then facilitating tick invasions? So are invasive plants altering forest communities in ways that are then making them more hospitable for ticks to establish in those places and bring pathogens with them? So is one of the reasons tick-borne disease risk is expanding, because non-native plant species are expanding in distribution as well. So as an overarching question for my talk, one of the things I'm interested to discuss with you is, are plant invasions altering tick-borne disease risk? And we often think of associating ticks with vegetation, so I think a lot of people can intuitively connect with this. Here's a picture of a black-legged tick questing from a stalk of vegetation. So we know for many tick species, vegetation is part of their host acquisition behavior. They climb up onto vegetation, they sit and wait passively for a host to come by, and they grab on when they get the chance. But ticks are likely interacting with vegetation in a number of other ways as well. So you may have learned from some of the presentations you've heard as part of this Tick Academy. Ticks, in many cases, spend a large portion of their life cycle off hosts. And when ticks are off hosts, they're experiencing a phenomenon called desiccation, which means they're basically drying out. And so what type of vegetation they're in and what type of local climate that makes for ticks might really affect their survival over time. And so that would be, as a mechanism, something that's affecting the habitat suitability for the ticks themselves. And so our non-native plants creating habitats that are more or less favorable for tick survival when they're off hosts. We also know there's several other players in these systems. When it comes to tick-borne disease, wildlife play a major role as well. They serve as blood meal hosts for ticks, so they're the source of nutrition that ticks depend on to complete their life cycle. In many cases, they're also the reservoirs for the infectious agents that the ticks pick up when they feed on wildlife hosts and then can transmit to humans. And so hosts play a major role. And of course, hosts are responding to their environment as well. And so non-native plants may change the quality of the landscape for the tick hosts, just as they might change the quality of the landscape for ticks. And then something that, to my knowledge, nobody has measured yet, but is a question I'm also interested in, is does it in any way affect how people get exposed to ticks? And so do people relate to invaded versus uninvaded habitats differently? And are there changes in human activity or human behavior that affect their exposure risk when they go into nature? And so I haven't seen anything published on that third question yet, excuse me, but I'll show you some evidence related to the first two questions today. So I'll start with the first project I ever did on invasive plants and ticks, which was looking at a very widespread problematic invasive species called bush honeysuckle. This is a genus of non-native plants in the genus Lonicera. Where I was working, the most common species was Amur honeysuckle. And this is actually a photo I took myself. I was living in St. Louis, Missouri at the time, out in kind of a wooded area. And everything you're seeing in the understory here is bush honeysuckle. One of the many features that can make invasive species successful is that they have some attribute that's different from the local community. In the case of bush honeysuckle, one of these attributes is they have what we call extended phenology, meaning they leaf out in the spring before most other plants, and they hold their leaves into the fall longer than other plants in these invaded habitats. And so this is a picture I took in the fall, just a few weeks later than where we are right now, of bush honeysuckle in the understory of this oak hickory forest in Missouri. And you can see the trees have all dropped their leaves already, but honeysuckle is still green. It's still photosynthesizing. And so it's got a longer growing season than all the native plants that it's displacing in this landscape. And so I was working in the St. Louis area at the time. I was studying Lone Star ticks, which are really prevalent in Missouri and Arkansas and throughout the Ozark ecosystem, where they transmit a number of diseases. And it was really striking to me, working across this kind of urban to rural landscape, how widespread honeysuckle invasion had become. And in fact, I was seeing it from the urban end of the land use gradient all the way out to the rural end of the land use gradient. I see a nice post in the chat here about military bases, and I'll actually show you some data from military bases a little later in my talk. So that's going to align well with your comment, Ed. Thank you. So here's a land use map for the St. Louis area. So here is like downtown St. Louis right here, if you've ever visited this part of the country. And so this is what ecologists refer to as an urban to rural land use gradient. And so in kind of these magenta-like tones over here, this is all urban land cover. But then as you head out of the St. Louis area, this all turns into more forested and more rural or more agricultural lands up here. And in this landscape, I identified a number of public parks where there were ongoing honeysuckle invasions. And across these sites, I did tick sampling. And I specifically selected these sites because they weren't fully invaded yet. So there were areas with honeysuckle and areas without honeysuckle. And in each of these places, I established experimental plots where I did sampling for Lone Star ticks. And my preferred method for sampling for Lone Star ticks are using these CO2 traps that you see in these images here. This is really simple technology, and this is part of what's fun about being an ecologist is some of the tools we use don't have to be especially sophisticated. What this is a lunch cooler attached to a plywood board. Around the outside of the plywood board, I would attach double-sided carpet tape. And so it had a sticky side facing up. And then I would put dry ice inside the coolers. Dry ice is frozen carbon dioxide. And as it sublimates, it turns into gaseous CO2. The gaseous CO2 pours out of the trap. And to a tick, it's one of the signals they're responding to that there's an animal host nearby, because of course we all breathe out carbon dioxide. And so when the tick smells carbon dioxide, it moves towards the trap and it gets caught in the double-sided tape. Lest you doubt the efficacy of this method, here's one of my more productive CO2 traps. So here you can see the double-sided tape all the way around the cooler, and every sort of brownish dot on there is a nymph or adult life stage Lone Star tick. So we would set these CO2 traps out in these areas, and we were using CO2 traps for Lone Star ticks because they're aggressive hosts. They're aggressive ticks. They actually will respond to a heat or carbon dioxide signal from a host, and they'll actually crawl towards that animal. So they don't just passively quest, they actively host seek. And because of that, you can sample ticks using this method. So putting these CO2 traps out in invaded and uninvaded habitats, we got this really strong signal from across these sites spanning the St. Louis area that areas with honeysuckle had much higher tick-borne disease risk than nearby areas that were uninvaded. And so in this first bar chart I'm showing you here, native vegetation on the left, bush honeysuckle or Lanicerum mackii on the right. And then what you're seeing on the vertical axis is the number of infected nymphal life stage ticks collected per trap. And what you can see is we saw a couple orders of magnitude higher level of densities of infected ticks in the invaded habitats compared to the uninvaded habitats within these sites. We suspected that one of the main drivers of this were how white-tailed deer were responding to invasions, and this was based on work we'd been doing up to that point, which demonstrated that white-tailed deer are both a key host for the Lone Star tick and also a key pathogen reservoir for the pathogens transmitted by the Lone Star tick. So we think white-tailed deer are playing a really large role in these disease systems. Fortunately, again, low sophistication technology, deer are very easy to survey for their relative abundance by counting their dung clusters. And so we can go out into the environment and count the number of dung clusters within a site, and it gives us some relative estimate of deer activity within a site. And again, we found that within the invaded plots, the density of deer dung clusters was much higher than compared to the native vegetation plots. And so we think this is likely one of the main explanatory factors for why we were seeing higher densities of infected ticks in honeysuckle areas, is that deer were being attracted to honeysuckle for some reason and spending more time there. We since did some follow-up work to try to tease out what we think was attracting deer to honeysuckle. We actually don't think deer are consuming honeysuckle in any large amount, and so probably they're attracted to it as a refuge because it creates this kind of dense structure within the understory. It's a place where deer are essentially choosing to hide out during the day, and so they're bringing their ticks with them and increasing tick abundance locally in honeysuckle invaded habitats. So our next question was, well, what if you remove honeysuckle? Because honeysuckle is one of the really noxious invasive species that is often targeted for invasion control. And so our next question became, if you remove honeysuckle, do you reduce tick-borne disease risk? And so we chose one of the sites within the St. Louis area. This is Bush Memorial Conservation Area, managed by the Missouri Department of Conservation. And we chose it because it had a high degree of honeysuckle invasion, a high density of ticks, a high density of deer. So it seemed like a really good place to ask this question. And then we got teams of volunteers to help us perform an experimental removal of honeysuckle. And so we, across replicated plots, removed honeysuckle. And then again, we compared tick abundance and deer abundance between plots where honeysuckle had not been removed and plots where honeysuckle had been removed. And this is my colleague Umberto Dutra, who did a lot of the design and actual work behind this restoration experiment. And so I really like to give full credit to Umberto for how much work this was to remove all that honeysuckle. And the experiment really kind of mirrored what we had seen in comparing honeysuckle invaded to honeysuckle uninvaded areas in other parks. We found where the honeysuckle was intact, the density of ticks infected with pathogens was much higher than where we had removed it. And the magnitude of these relationships are very comparable to what we saw in our surveys. And so we think removing honeysuckle essentially puts disease risk back to where it was before honeysuckle invaded, which was a really exciting finding. Again, we think there's evidence that the main driver of this is how deer are responding to honeysuckle invasions. In the plots where we removed honeysuckle, there were many fewer dung clusters per plot from deer than where honeysuckle was left intact. And for this survey, we also used a molecular tool to confirm these results. This is a technology that we developed around the same time, which was to actually take ticks that we had captured in nature and screen them for the DNA from the wildlife host from which they had fed. It's a really complicated technology and it's hard to make it work, but we were working with a fantastic molecular scientist at the time named Lisa Gessling, and Lisa was able to pull this off. And so analyzing the host DNA in these ticks, we found that in plots where honeysuckle had been removed, there was a trend towards those ticks having had fewer blood meals from deer compared to plots where honeysuckle was intact. Not quite statistically significant due to a small sample size, but I think the trend is supportive of the idea that the reason that we're seeing these responses of ticks to honeysuckle is because of how deer were responding to honeysuckle. We also had one other hypothesis for why honeysuckle might be altering tick-borne disease risk, is that it might somehow change habitat suitability for ticks as well. And so in the context of this same experiment at Bush Conservation Area, we also did a tick survival study. So we took adult and nymphal life stage Lone Star ticks, placed them in mesh bags, put those mesh bags inside wire enclosures, and distributed them between the honeysuckle intact and honeysuckle removed experimental plots at Bush Conservation Area. And then we monitored the rate at which those ticks died. And so that's called a survival study. And you analyze the data using a Cox regressional analysis. And what you can see is that for both the nymphs and the adults, there was no significant effect whether they were placed in honeysuckle intact or honeysuckle removed plots. They essentially died at the same rate. And so we would check these ticks weekly to determine the number of ticks still surviving. It's actually really easy to do. You just pull out the mesh bag and you breathe on it. And if the ticks are alive, that carbon dioxide signal will kind of wake them up a bit and they'll move and you'll know they're still living. And if they don't, then you record them as dead. And so this experiment ran for 22 weeks. And over the course of 22 weeks, the ticks really desiccated at very equivalent rates in the honeysuckle invaded and the honeysuckle removed plots. And so in this instance, we don't think there was any difference in tick survival due to honeysuckle invasion. Rather, it looks like it's entirely driven by how deer are responding to honeysuckle. And so we published the results around 2010. And here's just some of the same data I already showed you, just condensed into a single figure with multiple panels. And so in this case, we found that bush honeysuckle invasion was promoting tick abundance by concentrating wildlife hosts in invaded areas. But it's a pretty happy story in the sense that we can point out that, look, if you remove honeysuckle, disease risk pretty much goes back down to where it was before the invasion occurred. And so one could call this a win-win scenario where management of invasive plant species actually has an added benefit of reducing tick-borne disease risk. And so that was a pretty neat story to be able to tell at that time. What was interesting, though, is I had several other colleagues around the country who at this same time had also converged on this question of how are invasive plants impacting ticks. And so for the next couple of slides, I want to show you some of the results, not of my own work, but work of my colleagues that were being done in different places with different tick species and different invasive plant species. And you can see that this was sort of a moment in time where a number of us had converged on this question. And the differences between what we found, I think, is what's particularly interesting. So I had some colleagues at Indiana University who were working on some similar questions. This is work by Dave Civitello, Luke Flory, and Keith Clay, who were all in Bloomington, Indiana at the time. And in that part of the country, they were seeing a widespread invasion by an invasive grass called stilt grass, or Microstegium viminium. And Microstegium forms these dense monocultures in the understory of eastern deciduous forests. And their group was interested in whether this change in understory composition, which was so striking, could be impacting tick survival. And so they conducted a tick survival experiment. They used two different tick species, the American dog tick and the Lone Star tick, both of which occur in Indiana. And they placed them in plots of stilt grass and plots of a comparable native vegetation species. And then kind of like what I did with honeysuckle, they tracked the number of ticks alive over time. And in this case, they found that both tick species survived longer in the native vegetation than in the invasive vegetation. And so in this case, the invasive species appears to reduce tick survival and by extension could potentially reduce tick-borne disease risk. The mechanism by which they suspect stilt grass was actually reducing survival. In this image here, you see stilt grass when it's in its growing period, when it's nice and green, but actually in the middle of the summer, stilt grass goes through a period of senescence where it turns brown and it's no longer photosynthesizing. And that's the period of time that overlaps with when ticks are active. And so to recreate those natural conditions, they performed their study at that time. And so at that time in the summer, stilt grass appears to be really poor habitat for tick survival. And so at least during that period of time when midsummer, when Lone Star ticks and American dog ticks are active, stilt grass appears to reduce their survival compared to native vegetation species. So a very different story in this case concerning stilt grass and its impact on ticks. Meanwhile, in the Northeast, my colleague Scott Williams in Connecticut was studying another invasive plant species, a shrub called barberry, and looking at how it impacts the biology of the black-legged tick and its transmission of the pathogen that causes Lyme disease. And they found that barberry invasion increases the prevalence specifically of infected ticks. And it's a really interesting story. In the case of these barberry invasions, barberry appears to be really suitable habitat for white-footed mice. White-footed mice are one of the critical wildlife hosts in the Lyme disease transmission cycle. They feed juvenile ticks, and they're a really good reservoir for the Lyme disease pathogen. What Williams' work has demonstrated is that there aren't more mice in barberry than outside of barberry, but mice prefer to spend time in barberry, likely because of the structure and the protection it affords. And so mice are concentrated in barberry habitats. More juvenile ticks encounter mice in barberry than outside of barberry. And because mice are a good reservoir for the Lyme disease pathogen, those mice hanging out in barberry invaded habitats infect a lot more ticks than occurs in uninvaded habitats. So in the case of barberry in Connecticut with white-footed mice and black-legged ticks, there's an increase in the number of infected ticks due to a concentration of mouse activity in invaded habitats. So 3 different studies around the same time, 3 different locations, 3 different invasive plant species, 3 different groups of ticks, and we got 3 different answers. And so to me, this is really fascinating because it shows there's no simple story about how plant invasions impact tick-borne disease risk. And it sort of supports my tagline that ecology is complicated, and we have a lot more work to do to understand how plant invasions are impacting tick-borne diseases. So this takes me to some of my more recent work on this topic. And so I'm going to show you some of the work I've done more recently on plant invasions. And of course, plant invasions aren't occurring in a vacuum. They're occurring against a backdrop of other forms of environmental change. And so this is actually work that was funded in part by the Department of Defense through their Strategic Environmental Research and Development Program, or SERDP. And SERDP funded my group to work on military installations to study how climate change, fire management, and plant invasions are impacting tick-borne disease risk. And so that sounds like a lot, and I'm not going to talk about it all today. But if you look at this diagram on the right here, it kind of captures all of these arrows that we think are potentially at play. So real biological relationships between things in nature. We know climate change is affecting the use of fire management as a tool. It's also contributing to some plant invasions in some cases. As I'll show you in a moment, there are strong interactions between fire and plant invasions, particularly for the invasive plant species we studied in the southeastern US. These phenomena can impact ticks directly by changing habitat suitability for ticks, but also indirectly by changing wildlife activity or wildlife abundance. And so you kind of have this network of direct and indirect effects that are all playing out in nature. Actually, the last time I spoke to the Tick Academy, I mostly presented my research from this project on fire management. And so it's kind of fun for me to come back today. And now I'll present the flip side of it, which is the work on plant invasions. But again, as I'll show you in a moment, you can't fully decouple that from fire because those two things are really related to each other. So in the Southeast US, we chose to focus on another invasive non-native plant that's of particularly high interest to the Department of Defense, which is Kogan grass. And Kogan grass is a fast-growing grass species from Asia that is invading the southeastern US. And it's particularly problematic on military installations because it alters fire dynamics in significant ways. So it invades the understory of southeastern pine forests, and it's incredibly flammable. And that's really a problem for the high density of military installations in the Southeast US, where there's a lot of munitions training and other types of activities that can start fires. And by having a really flammable invasive plant species in the landscape, it really increases the chances of wildland fires occurring on military installations, which of course the Department of Defense wishes to avoid. And so we focused on Kogan grass. And here you can see some southeast pine forests, one invaded by Kogan grass on the bottom, a more species diverse one consisting of native species on top. These are naturally fire-prone ecosystems. So there's trees like longleaf pines that are actually dependent on fire for germination. And so these are fire-adapted systems. But Kogan grass comes in, and as you can just see from these images, among other things, it creates a lot of fuel. And once it goes senescent, that is all standing dead biomass that's highly, highly flammable. And so you can imagine a mortar shell or something else that could start a fire lands in that landscape. And there's a lot of flammable material there. In fact, as part of our research, we did experiments just to document the flammability of Kogan grass. And so you can see on the left, which is experimental fire involving native vegetation, and on the right, an experimental fire involving Kogan grass. Not only is there more fuel because Kogan grass creates a bunch of dead standing biomass, but it's also super flammable. The oils in the grass are really flammable. So it creates much more intense fires, which have the possibility of destroying mature trees, which is not typical of fires in that landscape. So we worked across 9 Department of Defense installations spanning the southeastern US that had varying degrees of Kogan grass invasion as well as fire dynamics. This is also in the range of the Lone Star tick. So most of the work that we did in this region focused on Lone Star ticks, but we're also in the range of the American Gulf, the Gulf Coast tick. And so that was another tick species that was present in this study. But most of the data I'll show you today will be from the Lone Star tick, which was the most common tick that we encountered across these military installations. In order to perform this sampling, we used a plot design that's known as a modified Whittaker plot, where we dropped randomized plots in invaded and uninvaded sites within these military installations. And within those, we collected a variety of data about tree density, understory vegetation. We again used our method of CO2 traps for sampling tick abundance, because both the Lone Star tick and the Gulf Coast tick will actively host seek. So this works really well by baiting these with dry ice. We had to modify them slightly because we discovered that in Florida, it rains every day in the afternoon. And so we had to put these sort of rain shelters over the traps so that the tape didn't get wet. But with that modification, we were able to use our usual method of tick surveys. And then within each of these plots, we also performed dung surveys to measure the abundance of wildlife hosts, not just white-tailed deer, but a number of other wildlife host species that occurred within these sites. Kind of as we expected, Kogan grass is actually a fire-prone invasive plant species, so it seems to do well in places that burn. And so looking at the number of years since a fire had transpired in a site that we sampled, we found that the more recently burned sites tended to be more invaded. So there's this connection between invasion and fire that's otherwise hard to get away from. And so from this point forward, I'll focus on the invasive plant data, but it's interesting to think about the connection between fire and these invasions. So the first thing I'll present to you is that we performed a tick survival assay akin to what we did with honeysuckle, although this one was more detailed. And we did the same strategy of putting ticks in mesh bags. These were both nymphal and adult life stage Lone Star ticks. And this time we also put out temperature and humidity data loggers. And that was so we could collect additional information on what were the local climate variables that were likely impacting ticks. If you're not familiar with the pattern of temperature and humidity throughout the day, temperature tends to peak in the middle of the day and humidity tends to be at the lowest in the middle of the day. And then it's the inverse at night. So temperature and humidity basically follow opposite patterns over time. And what we found is that the adult and nymphal life stage ticks survived a lot longer in Kogan grass invaded sites than on invaded sites. And so Kogan grass about doubled the survival time of Lone Star ticks that were placed in these enclosures. And we think the reason is because Kogan grass was really buffering ticks against the most extreme weather conditions they experienced throughout the day. So what you're looking at on these bottom two graphs is over the entire timeline of this survival assay, which took almost a year. So almost a year of having to go check these enclosures until all these ticks had died, especially in the Kogan grass plots where they really held on for a long time. You can see 80% relative humidity is something that's been identified in the literature as about a threshold for tick survival, where once it's below 80% humidity, ticks are really desiccating at a higher rate. And so all that time you spend above 80% is really kind of ideal conditions for ticks. And what you can see, the red, again, corresponds to Kogan grass, the blue corresponds to native vegetation. The ticks in Kogan grass over the course of the survival study were experiencing better relative humidity conditions throughout much of the study. Likewise, the Kogan grass was buffering temperature as well, and so the ticks in Kogan grass were experiencing lower temperatures compared to the ticks in native vegetation. And so Kogan grass seems to really be providing a more favorable microclimate for tick survival compared to the native vegetation in these sites, which were things like wiregrass. We also performed a statistical analysis. So these are some of these same data presented a little differently, which is looking at the number of days survived for nymphs and adults with the number of hours per day under 80% relative humidity, and then looking at that relationship with tick survival. And what you can see in these top two panels here is that over time, the relationship between the number of days they survive and the number of hours per day under 80% relative humidity is a negative relationship for the ticks in the uninvaded habitats. In the ticks in the invaded habitats, there's basically no relationship. And so you get a flat line. And so because they're experiencing less time below 80% relative humidity, there's really no relationship with the number of days they survived. Similar concept with the max temperature at the time of tick mortality, especially if you look at adults, there's a strong negative relationship between the number of days they survived and what was the max temperature at the time they died in native vegetation, but seemingly no relationship in the invaded sites. And so it really looks like Kogan grass is buffering these really extreme conditions that would otherwise be experienced by ticks in these sites. They're just spending less time at these extremely low relative humidities or extremely high maximum temperatures compared to ticks that were placed in native vegetation. We also performed host surveys in these sites. And again, white-tailed deer were the most abundant host. Several other host species were detected as well, including eastern cottontail rabbits. Wild turkey actually serve as hosts for Lone Star ticks. But deer were the majority of the host abundance data. So what I'm showing you here is the relationship between host abundance and the number of ticks we collected on CO2 traps for nymphs and adults in native and invaded plots. And what you can see is that deer abundance really predicted tick abundance in the native plots. So as deer abundance increased, the number of ticks increased in native vegetation. Interestingly, we see that host abundance is actually much lower in the invaded plots, and there doesn't seem to be any strong relationship with the number of ticks. And so the number of ticks doesn't really increase in the invaded plots as host abundance increases. And so what this is essentially telling me is that the main thing driving tick abundance in the native plots is host abundance, but less so in the invaded plots, likely because we think tick survival is the main factor going on in the invaded plots. And so what's really cool about this is, let me draw your attention to this graph on the right here, is that when you actually compare the number of ticks we collected in the native and invaded areas using CO2 traps, there's no significant difference between them. We saw a comparable abundance of ticks in both native and invaded vegetation. And so had we just collected data on how many ticks there were in native vegetation versus Kogan grass invaded sites, we might have concluded that Kogan grass is having no impact on tick-borne disease risk because there's no difference in the number of ticks. In actuality, we think that Kogan grass is impacting tick abundance by two different mechanisms, and they're offsetting each other. And for that reason, you see no difference. And so I'll bring you to this conceptual diagram over here. We think Kogan grass is significantly increasing tick survival, which is therefore likely increasing tick abundance. But hosts appear to be avoiding Kogan grass invasion, spending more time in native vegetation. So Kogan grass does not increase host abundance, rather native vegetation does, which increases tick abundance in native vegetation. And as a result, the magnitude of these two changes are about the same, and they seem to offset each other. And so you see about the same amount of ticks in native and invaded sites, not because Kogan grass isn't changing things, but because it's changing two things in opposite directions. There's more hosts in the native vegetation, which is likely the main driver of tick abundance in native vegetation. There's more tick survival in the invaded sites, which is likely the main driver of tick abundance in the invaded sites. And so this was a new one for me. I had never sort of seen multiple things changing at once, and then the net outcome was no difference. And so that was really interesting to unpack. And for that reason, I'm glad we collected as much data as we did, because I don't think we would have understood the ecology of this particular instance if we hadn't done so. So now that's 4 different studies, 4 different locations, 4 different invasive plant species, multiple different tick species. And again, we see differing results in all cases. I've shown you 2 examples of where plant invasions have increased tick-borne disease risk. One example now from Florida, where a plant invasion does not change tick-borne disease risk, but only because it changes multiple things at once. And the example from Indiana, where an invasion appears to reduce tick-borne disease risk. And so what we're learning is that disease risk can increase, decrease, or remain the same depending on the mechanistic pathways that are involved in nature. But clearly, plant invasions can alter tick-borne disease risk to humans and wildlife. And so I think quite a bit more work needs to be done to understand these mechanistic relationships. Some of these tick species have wide ranges, and so the relationship between ticks and honeysuckle in Missouri could be very different than the relationship between the same tick species and the same invasive plant species, but in a different part of the country. So we have a lot of work to do to kind of unpack all those relationships. That said, I do think there are going to be a number of examples where invasive plants are increasing tick-borne disease. And like what we saw with honeysuckle, the removal of those invasive plants may also have the added benefit of reducing tick-borne disease risk. And so I think there are going to be some win-win scenarios out there where controlling invasive plants also will have the added benefit of reducing tick-borne disease risk to humans. And I think that makes for a really strong management argument that those are places where controlling invasive plants have these additional benefits. I want to especially thank the project team from the CERTUP project, because that was a really large project. So here are the many field assistants who assisted with that work. The postdoc who ran it on the left, Waylon Dillon, and the laboratory scientist who did all the laboratory work on the right, Paige Fredericks. I also want to acknowledge my collaborators from University of Florida, Boston University, and my own lab. Here's our very first tick trap with a rainout shelter. We were really proud when we finally figured out how to keep these tick traps from getting wet. Thank you. I'm happy to answer any questions, and I hope you enjoyed my presentation.